Today, we're diving into a really revolutionary new weapon against sepsis. It's basically a microscopic, programmable smart bomb designed for our own immune system. And it is a completely new way of thinking about one of medicine's biggest and deadliest challenges. But before we get into the tech, we have to start with a tough question. How in the world do you stop an army that has started attacking itself? Because when you boil it all down, that's exactly what's happening with sepsis. You can think of sepsis as a full-blown civil war raging inside your own body, and the stakes just couldn't be higher. It's so incredibly difficult to treat because the enemy isn't just some invading bacteria, it's your own defense system going haywire. And this right here is the most critical thing to understand. Sepsis isn't actually the infection, it's the body's catastrophic overreaction to it. The immune system just goes into complete overdrive, and its response becomes way more dangerous than the original bug, causing organs to fail and all too often, death. Now, deep inside our immune cells, there's this key player that's responsible for sounding the alarm. It's a protein complex called the NLRP3 inflammasome. The easiest way to think about it is like a super sensitive inflammation alarm system. The second it senses danger, it pulls the fire alarm and triggers a massive defensive response. And normally, this system is a lifesaver. It's what you want. But with sepsis, that alarm gets jammed in the on position. It just keeps screaming, danger, danger, long after it needs to. And this leads to what you can only describe as catastrophic friendly fire, where our own immune cells start attacking our body's healthy tissues and organs. So the big question is, how do you turn off a faulty alarm that's buried deep inside thousands of individual cells? Well, researchers have come up with a brilliant solution, a brand new programmable medicine. So the whole strategy was basically to build a microscopic smart bomb, and it had three main goals. First, it had to be sneaky enough to get past the body's defenses. Second, it needed to find the exact immune cells with that broken alarm system. And third, it had to shut that alarm off, but, and this is key, only when a doctor gives the order. Okay, so let's break down how they did this, starting with the first part of the mission, creating the vehicle itself. I mean, how do you even begin to build a tiny machine that can get around inside a body that's already on high alert? This is a huge challenge. The scientists are using a modified virus to carry their therapeutic tool. But in a patient with sepsis, the immune system is hypervigilant, it's actively hunting and destroying any foreign thing it can find. A normal therapeutic virus wouldn't stand a chance. The solution they came up with is just so clever. They decided to give the virus a stealth cloak. They used a process called biomineralization to coat the virus in this thin, protective shell made of calcium phosphate. It's a totally natural mineral that the body recognizes, so it's way less likely to get attacked. And you know, the process itself is surprisingly simple. You just start with your engineered virus, you add some basic calcium and phosphate ions into the mix, and almost like magic, a mineral shell just crystallizes right around the virus's surface. What you're left with is a stable, camouflaged particle, totally ready for its mission. And check this out. These images from the electron microscope really show you the transformation. On the left, you've got the naked, vulnerable virus. And on the right, you can see the final particle, totally disguised in that smooth, protective mineral shell. Pretty cool, right? Okay, so the vehicle has its stealth cloak, but how does it know where to go? It can't just go around affecting any old cell. It has to be incredibly precise. The therapy needs to target only the specific type of rogue immune cells, these attack cells called M1 macrophages, that are causing all the damage. The solution here is to add a kind of homing beacon to the outside of that mineral shell. What they used was folic acid. It just so happens that these super angry M1 macrophages have a special receptor on their surface that folic acid latches onto perfectly. It's like putting a very specific key on your delivery truck that will only open the locks on your target's front door. So we have a stealth vehicle and a targeting system, but now for what might be the most brilliant part of this whole thing, a remote control on switch that gives doctors total control over the therapy. This is such a critical safety feature. You're delivering a really powerful tool that can switch off a key part of the immune system. So how do you make absolutely certain that it only goes active at the exact right time? Well, the answer is in how they designed the therapeutic tool itself. It's an RNA editing system called Cas-Rx. The researchers actually engineered it to be unstable on purpose. So by default, the tool is kind of like a machine with a bunch of loose parts. It just falls apart inside the cell really quickly and becomes completely useless. But here's the genius part. A really common, safe antibiotic called trimethoprim, or TMP, acts like a molecular glue. When a doctor gives this drug to the patient, it goes into the cell, finds the tool, and locks all its parts together, making it stable and functional. It literally turns the therapy on, and when the drug naturally wears off, the tool just falls apart again, turning the therapy off. It's a perfect controllable switch. And here is the undeniable proof that it actually works in a living animal. The system was designed so it would produce light whenever it was active. So before the drug is given, the mice are dark. But after they get the drug, you see the target area light right up, showing the therapy is on. Then, as the drug clears from the system, the light fades away, confirming the system has switched itself back off. So the engineering is just brilliant. We have a stealthy, targeted, and controllable system. But the most important question is still out there. Did this amazing piece of technology actually work against sepsis? Let's take a look at the results. And the results are, well, they're nothing short of stunning. This simple bar chart tells you everything you need to know. In the mouse model of sepsis, the group that got no treatment had a 0% survival rate. But the group that got the new programmable therapy, an incredible 80% survival rate. And the treatment didn't just help them survive, it prevented that awful organ damage. On the left, you can see these microscope images of lung tissue from an untreated mouse with sepsis. It's totally inflamed, filled with destructive immune cells. But then on the right, the lung from a treated mouse looks clear and healthy. The therapy literally shielded the organs from all that friendly fire. So in the end, the mission was a complete success. This programmable medicine got to the right cells, it turned off that faulty alarm system, and it calmed down the immune system's overreaction. And by doing that, it saved lives and prevented the collateral damage that makes sepsis so incredibly deadly. And this really leaves us with a fascinating final thought. Okay, so sepsis was the target here, but this technology, a therapy that you can cloak, that you can target, and that you can switch on and off with a simple drug, this is a platform. It really makes you wonder, what other diseases, the ones driven by specific cells going rogue, could we treat with medicine that we can truly program and control?